So, welcome everyone. Thank you to the organizers. It's a fantastic symposium. And of course to everyone that stayed to the last session. So I think it was quite crafty of the organizers to put sun research and plant research together. And it's because one of the most magical things about plants is that can actually harness sun energy and convert it to chemical energy, which basically means to sugars. And in the process, which is called photosynthesis, they generate oxygen and they generate sugars. And without sugars and oxygens, we would not be sitting here. So it's a really magical process. And this is also one of the processes that is studied heavily in my institute, which is located in Golm. And we already heard about Golm today from Daniel. So there's nothing particularly special about Golm, besides maybe a very high density of researchers per square or a square kilometer. But it's also super nicely located because it's close to Potsdam, which is a really beautiful German town, famous for its parks and palaces, and also very close from Berlin, which is a German capital and a cultural center. So this is institute itself. So here would be the colloid interfaces we've heard today about. This is plant biology. So we have 3 floors and 3 departments, about 20 research groups. We also have many tandem groups in Latin Americas, and also one here in Bogota. So where is Federico? Hey, Federico. So Federico actually visited our institute about a couple of years ago, and we're really impressed by his research plans, and I'm really happy to see him again, and I hope he will visit us again soon. So the very general kind of interest and a broad interest of our institute, and besides photosynthesis, would be plant physiology and plant growth physiology, and also metabolism. So to study plant growth, obviously you need, or to study plants at all, you need greenhouses, and we have fantastic greenhouses. So kind of the newest fashion in plant biology is to bring everything close to field condition, for example, by having polytunnels like that, where plants experience natural light conditions and they're exposed to night and day cycles and normal temperatures. Whilst to study metabolism, the institute invested heavily in this kind of machines. They're called mass spectrometers, and you know, we are so fond of them that we actually give them names. And just today my postdoc emailed me that our favorite mass spectrometer, Yvonne, is very sick. Poor Yvonne. I hope she will recover. So these instruments, basically, they allow us to measure mass and charge of chemical molecules, and in that way identify and quantify things that we call metabolites. So very kind of simple introductions. What is metabolism? Is basically all the chemical reactions involved in maintaining the living state of an organism, and this is just, I've actually, I think, seen a snapshot of that already today. So it's basically kind of a simplified map of central metabolism. So what is metabolite, or a small molecule? So the central focus of my talk. So basically, those would be substrates, intermediates, and products of metabolism. So I give you some examples. So for example, sugar is a metabolite, and it's an energy source. An amino acid is also a metabolite, and it's a building block for proteins. But also neurotransmitters like dopamine and serotonin, they're also metabolites. And they're also kind of the newest that took craze, apparently. And then we have, you know, this is basically a vial with a substance called vinblastine, which is a drug used for cancer. And this is a metabolite produced by this plant called Catharanthus. So you have a metabolite, it's a plant metabolite. We don't really actually know what is the function of this metabolite in plant. And it happens to be a very important small molecule because it's a drug. Actually, plants also produce these neurotransmitter-like compounds, but I mean, they obviously have to have a different function than neurotransmitters. Again, we would not know what this function is. And this brings me to kind of a very important thing. So plants as microorganisms that we already had today are fantastic organic chemists, and we don't fully understand like even the full complexity. We don't know how many metabolites exactly plants produce, but it's clearly in thousands. So many of unknown nature and even many more of unknown function, the function in plants itself, like, for example, those and also vinblastine and vincristine. So this is, again, something that was already discussed today. So for a metabolite to function, it has to interact with something. And most often it would interact with a protein or macromolecule. So basically, when you think about cells and processes, you have this very kind of complex, intricate networks when things interact with each other, biological entities like metabolites and proteins and RNAs. And this is very kind of similar to social networks. So everything has to be connected. And this is really not giving the justice because those connections would be extremely dynamic. And this is obviously a very static and a very limited picture. So this brings me to the goal of my group and my research. And this it would be to identify metabolite functions. And because metabolite function by interacting with proteins, we thought, well, if we figure out the interaction partners, the protein interactions partners of our metabolites, maybe we'll be able to say something about the function, kind of in a way that maybe you try to guess a character of a person by looking at the friends of this person. And the important thing is that we know much more about proteins than we know about metabolites. I will skip that, anyway, it's a bit cut. So how to study interaction? So kind of our expertise is really in mass spectrometry, in biochemistry, so this was kind of the obvious thing for us to do. But when you, many of the kind of biochemistry we do, we don't, what we need is we basically grind the cells, right? Like we, it's a very disruptive process, and we have also lengthy biochemical separation. So we kind of wondered how many of those interactions that happen in the cell will survive the process. And to test that, we did this kind of very simple experiment. So this is called a kind of a size filtration. And you have this kind of a filter here that has a size cutoff. And if you put this kind of a native lysate, so basically you grind, you open the cells, and all the content will be this native lysate. And this native lysate will contain complexes, but also free small molecules and unbound proteins. And you put it on this filter and you centrifuge. So all the small things, all the kind of free small molecules, they should go to a flow through. Whilst if you find something on the filter, it's there because it was bound to something bigger, bound to a protein. And we're actually extremely happy that we found many metabolites separating with protein complexes, telling us that what we are planning is feasible. And those would be primary and secondary metabolites, but also hormones, really a lot of peptides. Now we also know it actually also will work for drugs and agrochemicals. So from that, we had to kind of create a toolbox, and we, the group started 2015. I think now we are at the stage that we have all these methods available, and there is a number of ways we can access the interaction. So imagine that this is basically the content of the cells, and you have a complex here composed of proteins and small molecules, and here you have the free small molecules unbound. So one of the ways is when you actually interested in a particular protein, you can put kind of a handle, kind of a tag that you can pull your protein of interest together with all its interactors. Then you can also do the reverse. If you actually have a small molecule that you're interested in, you can put such a handle on a small molecule, and again you can pull complexes. And but we also kind of went further with the size separation idea, and we introduced this chromatographic step. So we not only separate what is bound from what is unbound, but we also separate complexes based on their size. And this is kind of our central approach. It's untargeted because you don't need to predefine a protein or a small molecule, and I will tell you more about it. So basically here you can start with any kind of biological sample. We work mainly with a model plant called Arabidopsis and with yeast. Then you prepare a lysate and you put it on this size separation column. And the size separation columns vary, and they can separate different complexes. You can actually also have columns that separate membrane complexes. And then we collect fractions, about 40 to 50 protein-containing fractions, and then we measure both metabolites and proteins in those fractions. So basically, here the assumption is very easy. If we find a small molecule in the protein-containing fraction, it was there because it was bound to a protein. And now we also have some nice collaborations, and we also tried it for, like a kind of agricultural plant for maize, but also actually for Catharanthus, and this is a collaboration with Sarah Corn in Jena. All right, so what you end up with is thousands of profiles, both for proteins and for metabolites, and often those would be very complex profiles, so we have to deconvolute them to single peaks, and then you basically correlate everything with everything, and here basically what colludes together can form a complex. And this is cut, so you will not see, but basically what was showing, what I wanted to show with those two things here, so two different data sets, one for yeast and one for plants, and we can see really, hundreds, if not thousands, of metabolic features separating with proteins, arguing for a really high complexity of the protein metabolite interactome. So what can you do with even like a single data set? So you can also actually query it for protein-protein interaction, so you get both in one experiment, protein-small molecule and protein-protein complexes. So this is just example of some macro complexes that we managed to isolate and separate from yeast. You can, of course, query it against known interactions to check how good is your method, and we retrieve about 5 times more of the known interaction than by chance, so it's okay. You can also query it against predicted interactions, predicted, for example, based on the homology, and again here we could experimentally validate about 1/5 of the predicted interactions. And some of those are actually quite interesting, and those would be novel kind of regulatory interactions. A lot of those allosteric regulatory interactions in the enzymatic pathways. So for example, here we could show that xanthine coalutes nicely with the upstream enzyme called PNP1, and we find an inhibitory effect. Okay, good. But what we are actually most interested in the group would be regulatory or signaling small molecules. And one molecule like that is abscisic acid. It's a plant hormone. It's very well characterized, and we're extremely happy to find it in our separation. And we also find a known interactor of abscisic acid, a hormone of abscisic acid, separating together with abscisic acid. So this is a nice validation. But then we also had this novel maxima here, and when we look what coalutes under this maxima, there was nothing that was previously reported. So this is of course something that we are extremely intrigued about. But the thing about this method is obviously the separation has its limits, right? So coelution is a kind of indication, but it's not yet a proof of interaction, because many things can coalute in the same fraction. So you still basically end up of, let's say, 200 proteins, and maybe only one is a true target. So here we always have to go with targeted approaches. And I'll just give you one example here. So this, so we are interested in this molecule. It looks very similar to the very well-known secondary messenger, but it's actually not. It's an RNA degradation product, and we saw it so nicely separating with proteins, so we wanted to know what it interacts with. Again, from this kind of promise data sets, we had too many hits, so we narrowed it down by this targeted approach called Affinity Purification, where you basically immobilize your small molecule on the agarose bit. So here we did kind of this fishing thing that I was showing in one of the slides. And this narrowed down the list to one protein only, which is great. And we could prove the interactions by a biophysical method called microscale thermophoresis. So this molecule, in fact, binds to our protein. And also, a very nice thing about this protein is that it's a very well characterized protein. So it's involved in formation of stress granules, which is one more example today of liquid-liquid phase separation body. And those stress granules are really important because they sequester RNA and proteins during stress, and inside the stress granules, the RNAs and proteins are being protected. And we could basically show that our molecule facilitates formation of the stress granules. So we know we basically started with this exploratory approach. We identify an interesting small molecule, and then we could narrow this protein target, and this led us to a really interesting biology. So we can also do the reverse. Rather than starting with a small molecule, we can start with a protein. So here again, just an example. It's not really important what this protein is. I mean, it's important for me. But what is important here is that basically we can put a tag on any protein that you're interested in, and we can pull the protein with its protein metabolite interactor. So previously, of course, people did a lot for the protein-protein interactions, and now basically we know that we can do the same for the protein metabolite interactions. So basically this is this PDF2 transcription factor, and this was the work together with Katrin Schrick from University of Kansas. And we got this really nice interaction network with both direct and indirect partners, and we're most excited by the presence of phospholipids, phosphatidylcholine, because this protein has a lipid binding domain. And together with Katrin and putting all extra genetic and molecular work, we now have this model where binding of a phospholipid to our transcription factor actually activates it. And interestingly enough, this transcription factor actually represses phospholipid catabolism. So we kind of think that we have a nice regulator that connects phospholipid sensing with phospholipid metabolism. So basically, I was showing you all these examples to prove that small molecule metabolite protein complexes can be studied using biochemical approaches. So you can do it in a very untargeted fashion by making these interaction maps, or a very targeted fashion starting with a small molecule or protein. And this actually applies to both metabolites but also drugs, which I haven't shown today. It also shows like an amazing complexity of the protein metabolite interactome, because we really get hundreds, if not thousands, of metabolic features separating with protein complexes. And I was trying to convince you that we really retrieve meaningful interactions, and that finding a protein target is a good start to functionally characterize a small molecule. So basically, I'm very happy with this methodological toolbox we have at the moment, and we can address really a lot of exciting biology with it. So one thing I'm kind of really interested in, and this comes from my background, is cell cycle regulation and the connection to metabolism and how metabolites play a role in that. But I will like to use my last 2 minutes showing yet another project I'm really excited about, and this is something I already also discussed with one of the tandem group leaders, with David Johnson, that was a chair today. So he gave me some very good advice here as well. So this project is a collaboration with a fantastic microbiologist from the Institute in Tübingen, Talia Karasov. So basically here, and also we've had it already today about it. So organisms don't live in a limbo, right? I mean, they are surrounded by all the other organisms, and many of those being microorganisms. So they basically all the time interact with each other. And some of those interactions will have no effect, but some will be deleterious, whilst others will be extremely beneficial. And it's exactly the same for plants. So we have microbes that basically cause devastation to crops, but then you have other microbes that actually stimulate plant growth and productivity. And one of the big things, and I guess maybe we'll also hear it about it tomorrow, is finding chemical signals, because it's basically the small molecules, the metabolites, that serve to communicate, the communication between the organisms is actually to find and characterize. I see the red light, but it's really kind of it's really 1 minute more. So here basically we thought, why not to study why not to identify those metabolites by studying protein metabolite or generating metabolite interaction maps in the function of infection, and then looking specifically for microbial compounds separating with plant protein complexes. And it does actually work, but I will skip the slide. I will skip this as well. And I would like to thank you, so my director, Professor Wilmitzer, but also amazing group members. One is actually Colombian. Many students. We also had a Colombian internship student, and we're extremely happy to work with him. So with that, thank you everyone, and I will be happy to take questions.